Thank you, David. Thank you so much. Such a nice and personal introduction, and I really appreciate it. And thank you all for coming this early morning. And it's really been a pleasure. I've been working with David for nigh on 30 years at this point. And I really want to thank ARVO for this honor, which is really very meaningful to me. I mean, I've always loved this meeting. It's the first meeting I came to as a young scientist, and I've continued all these years, and I'm just wonderful to be here and to see so many old friends. I'm particularly glad to be here in the same year that my two colleagues from the annual review of Vision Science, Jamie Wiggs and Rachel Wan, are winning awards as well. And it's a particular honor to be in their same company. So again, thank you very much. For the next 30 minutes or so, I'll be reviewing three related topics about the structure and the function of the human brain. All the slides that I show and the references therein will be available in a day or so on my website, and I hope they're useful to you at some point. So the first topic concerns what we've learned over the past 30 years concerning the organization of human visual cortex, and in particular the retinotopic maps and how they are organized together. The second topic builds on these discoveries to review what we've learned about the plasticity and stability of those maps. That was something that occupied us for quite some time. And the third topic will be how we're using these methods to understand how the visual signals are then passed on to important cognitive and emotional aspects of the human brain in our lives through, in particular, the focus will be on reading, in particular the plasticity involved in how children learn to read. So coming into each of these topics, they each already have, long before I ever showed up, a huge history that's really quite fascinating, and particularly the history of how we came to learn about visual cortex. I see John Robson in the audience, and I'm thinking of Sir David Ferrier, who engaged in quite a significant debate with Monk, shown here on this slide, about where visual cortex was. Is it in the back of the brain? Is it the front of the brain? How are we going to get this stuff right? And I would say it was even more intense than many of the Twitter wars that I've seen these days. This was the real, got quite personal. Fascinating story, but over time it evolved, and a lot was learned from Henshin, Inoue, and Holmes, so that by the 1920s, we actually had a very decent understanding of where visual cortex, primary visual cortex, was located, and the retinotopic map that was there. And the reason for this was that the bullet wounds from the wars in those days were non-lethal, and a lot of people would have injuries and little lesions or significant lesions in visual cortex, and by mapping those lesions, they could learn where it was they were blinded by the bullet that passed through their brain. After the 1920s, there's the discovery of a new generation of technologies so that people could make very precise measurements in animal models using electrophysiological measurements. And by the 1990s, we had a really pretty good understanding of a lot of what Dave Van Essen would call visual areas, some of which were maps. And this famous diagram here from Dan Fellman and Dave Van Essen is widely known and widespread to give a sense of these animal models. But in the human, and by the way, different animals had quite different organizations. And in the human, we had this very nice paper from my wonderful colleague Jonathan Horton and his colleague William Hoyt, who wrote that even in 1991, at the time of the Fellman and Van Essen diagrams, nothing was known about the topography of V2, the second visual map in human visual cortex. And in fact, in their work, they corrected the Holmes map. That was part of what they were doing. And why was that? Well, it was really because we had very limited access to being able to measure the human brain. In 1986, this is a kind of paper that would get you into nature, if you did a PET study where you had stimulated two different positions and you'd see a white spot move from here to there. You could write about it for a couple of pages and it was an exciting new discovery. But by 2010, and I want you to feel this, all this progress, because that's really, I felt lucky to be part of this. By 2010, John Winnower and I could write a summary paper of what had been learned and what the new techniques were capable of, and we could show you things like, well, this red region is V1 and surrounding it is V2 and V3 in individual subjects, individual people who we could study in great detail that way. And we could also measure the eccentricity representations or angular representations. These colors here, and this one, are now showing you the foveal representation to the near periphery, to the further periphery, in a single individual in the posterior part of their brain. So that's quite an advance in technology, and it really gave us the opportunity to learn a lot about the brain. And the basic idea of how you make those maps and that progress started with some simple experiments like the one shown here. A subject is staring at this red fixation. I'm showing you an inflated version of their right hemisphere. The dark regions are sulci, the lighter regions are gyri, and you're seeing the activation in visual cortex from this kind of expanding ring stimulus that was sometimes called a traveling wave stimulus for obvious reasons. And if you like to be more obscure, you could call it phase-encoded retinotopy. Those were your options. So we could represent which part of the visual field was particularly effective at exciting different locations in cortex, and that's shown here by this color map, where the foveal region, is represented here in the posterior part. It actually wraps around the edge, which was a surprise to many of us. And then near periphery and the far periphery here for the eccentricity map. And if you make the measurement in the other direction, instead of expanding on out, you measure the angles swinging around, shown with this stimulus over here. You can see that, in fact, the lower vertical meridian, as Henshin had shown us nearly 100 years earlier, the lower vertical meridian is represented on the upper bank and the lower vertical meridian on the lower bank. And here you have the retinotopic map for V1, and which could be clearly measured and easily defined. Okay, so that was great to go and confirm some of what had been shown by prior work, but there's really a lot more was learned in those years. So here's an example. I'll give you a couple. Here's an example. Notice that there's a region down over here that's also representing the fovea, and it's distinct. There's a gap between the foveal region in the very back of the brain and this region over here. It's another little eccentricity map. And and in fact, within that eccentricity map, there are again multiple submaps. If you do the angular one, you can subdivide it into different regions. And in fact, we came to learn that as you look through the human brain, here was the original map, V1 and others that had been found. Here's the ventral occipital map that I was just showing you a moment ago. And in fact, on the lateral aspect, this is both of these are the left hemisphere of one brain. You're looking at the lateral surface here, the medial surface here. You can see there's another eccentricity map headed out this way, and another one over here, and another one over here, and another one over here. So that visual cortex we've learned is structured into a set of clusters of maps, each with its own eccentricity representation, and each one can be subdivided into multiple visual areas. As we were learning this, we also introduced the current naming convention, in which the new maps and their positions that we were discovering were labeled by their positions, such as ventral occipital, lateral occipital, and so forth. And to those of you who are young students and don't have to go through the process that I went through to figure out what's TEO, MST, V8, V7, and so forth, all I can say to you is, you're welcome. Now, not everything in human cortex was a recapitulation of the non-human primate work. In fact, there are significant differences between all the primate species, and human differs from the others in a way. And I can't go into that in detail today, but I hope that those of you who want to follow up on that get a chance to look at the literature and see the differences, which are notable. The next step in this process, once we had the maps identified, was to try to understand what about the visual stimulus evokes a response in these maps and how the signals in these maps go together. And we built on, again, on the classic work. Here I'm citing Hartline, but also back to Sherrington and Kuffler, of course, of people who would make receptive field measurements, but using, in our case, we're using magnetic resonance instruments to measure the receptive fields of small regions of visual cortex. And so we could do that by literally sweeping stimuli, doing the same kinds of stimulus mapping experiments that were done with electrophysiology. But since these were not receptive fields of individual neurons, but groups of neurons, we call these population receptive fields. And that's what's shown here. And there are very big effects. This work, which I started with Serge Dumoulin some 15, 20 years ago, what we could show, for example, that in the medial part in calcarine cortex, the receptive fields are quite small. If you go to the lateral brain, where there's motion responsive cortex, the receptive fields are much larger. And within each of the maps, there are quite regular organizations as you measure from fovea to periphery, where the receptive fields would start out to be small and get bigger and bigger as you looked at different eccentricities. And these are the standard kind of conventional way in which people's brains develop, in which visual field maps arise. And so we have now a kind of a sense from these many different maps. Oh, I should say everything I've shown you up to this point has been replicated in multiple labs. These aren't edgy little findings. This is the whole field working together in a cooperative way to go and confirm one another's work, check one another's work. And there was a certain amount of pushing and shoving, but I say it wasn't as much as what happened between Ferrier and Monk. We were a lot more civil to one another. Okay, during this period, you're always looking for like, what's next? Okay, you're finding these maps and great, you're understanding what it is. But what is it that you might like to understand about these maps and how they develop? And we were, of course, during that era, quite engaged by the significant claims that built on the original work of Hubel and Wiesel, the Nobel laureates in vision science, who had shown these crucial ideas about developmental, critical periods of development. And people thought, well, okay, critical period is something that happens during development in young, but then after that, the brain is relatively stable. And at that time, in the late 90s and 2000s, Mike Merzenich and John Koss and others were saying, whoa, this is just not so. There's a lot of adult cortical plasticity. And so we were interested in this to see whether we could characterize it and measure it and so forth. And we started to take a look. So back in about 2000, a little past in 2002, with the help of colleagues from ophthalmology, I was fortunate to be able to recruit some congenital complete achromatopsic patients, or some colloquially called rod monochromats. And they were a great topic to try to understand some developmental plasticity. And the reason for that is, as most the very center of your gaze in your fovea, there's a region about 400 microns across, about 1.25 degrees of visual field, where there are no rods, what's called rod-free zone. And by the time that part of cortex that part of the retina is mapped into visual cortex, it actually is spread and occupies a very large amount. The central 1.25 degrees occupies a very large amount of cortex. And I'm showing you, here was the map I showed you a moment ago from fovea to periphery. Here I've computationally taken this hemisphere here and flattened that region so you could just see it all at once. And you can see what a large amount of area is actually representing the very, very central foveal part of the visual field. Well, if you take a normal subject and put them under scotopic conditions so that they're only using their rods, it's easy to see that there's a massive amount of silent cortex, just not responsive, even though right next to it where the rods are present, it is responding. So what happens when you take a rod monochromat who was born without any cones? Does this stay silent in this region of cortex? Does this just stay silent throughout their lifespan? And the answer to that was easy. The first 3 rod monochromats that we measured, no, not at all. The rod signals come up to cortex, and during development, they fill in that, they take over that part of cortex, and you can see it easily. And notice, by the way, that whereas in the control subjects on the right, you see that red part, for which is the central fovea. In the rod monochromat, the rods are in the periphery, and it's got the greenish part because they're in the periphery, but it's still causing that piece of cortex, the corresponding piece of cortex, to respond. Here was another kind of developmental plasticity that we could easily measure. Most of you know that the optic nerve, as it projects into the brain, the nasal portion of the retina that comes out of the optic nerve from the nasal portion crosses to the other hemisphere. So the temporal part stays on the same side, the nasal part crosses, and we get the usual hemifield representation in primary visual cortex. And you can see the crossing in magnetic resonance imaging quite easily. Well, one day I'm minding my own business, and a colleague of mine, Joyce Liao, calls me up from the ophthalmology department and says, look, we've got a subject over here who came in for headaches, but look at this. It seems kind of odd, doesn't it? And you can see there's no chiasm there. And we thought these cases were extremely rare. They are quite rare, but Michael Hoffman in Europe has collected up quite a number of them at this point. And so they said, but this is so strange because his vision is fine. He reads, he plays baseball, he doesn't have stereo vision, but he could do all kinds of other stuff. Could you have a look? And so, we did, in fact, block out one eye, and we put things in the contralateral hemifield, and sure enough, we get a response on the right hemisphere, and then we put things in the right hemifield, and we still get a response in the same hemisphere. So one eye in this person goes to one side of the brain, none of the crossing, none of the hemifield. Yet the person managed and did not come into the clinic for the purpose of having their vision checked, but it was just an incidental discovery. It was quite capable visually. I find this amazing that the brain during plastic development can have such a radically different organization, and people still, the rest of the brain manages to deal with the signal. So I thought that was amazing. We did also measure population receptive fields in this individual, and they're organized in quite a regular way, where one little piece of cortex sees the corresponding bits of the two hemifields. It was quite an interesting story from my point of view. So at that time, or just around that time, we thought, well, to get a better understanding of the molecular basis and the cellular basis of this, maybe we should try to make some measurements in animal models. And I was lucky to have a close colleague and good friend, Nikos Logothetis, and his postdoc at the time, and now a distinguished neurologist at Harvard, Stelios Smirnakis, and we spent years going back and forth getting rigs built to make measurements in macaque brain, again using these same functional MRI methods. And shown on the left side of the screen is a picture of us demonstrating that yes, you could make retinotopic maps in primary visual cortex of a macaque monkey, and the signals were fine with Nikos's rig, which was quite a feat to build that rig, so that was important. And then Stelios went on to try to understand the plasticity, this time in the adult macaque. So these were not developing cases, these were fully grown macaques, and what he did was introduce a lesion into both the left and the right eye, and so those corresponding parts in the typical eye will project into a zone into the macaque primary visual cortex called the lesion projection zone. And in this particular case, sure enough, you lesion the retina, and those portions of V1 are silent. So we thought, okay, great, now we're going to wait for this adult cortical plasticity to happen, and these things are going to fill in, and we're going to be able to watch that. But in fact, as we measured week after week and month after month, there was virtually no change in the magnetic resonance imaging response. And this was rather controversial, so that even though this was a nominal replication and a nominal attempt to see plasticity that had been reported in a certain way, the failure to find it was in fact accepted and published in Nature, and caused, let's say, a little bit of a back and forth over time in that. I was very lucky at that time to have two outstanding, well, actually a series, but two who are here, outstanding clinicians, ophthalmologists, and retinal surgeons who at that time were interested in this and took a risk and came to some random psychologist's lab to go and do their work. And in fact, in a series of papers that I won't go through in detail, but that were convincing to me, Yoichiro Masuda and Hiroshi Horiguchi are here, and they made measurements in juvenile macular degeneration cases where the lesions were natural from diseases, retinitis pigmentosa cases, and our rough conclusion about the stability of the maps that we had seen in the measurements with, in the adult, that we had seen in the cases with logothetis were confirmed, and so forth. There was one kind of interesting case that showed up where, in these cases, I've shown you that if you are born and early and it develops in a way that's quite unusual, you can learn to see. There are also in our field something that I find fascinating, these so-called miracle cures. And these are cases in which maybe you have a disruption at birth and you don't see very well, and then later in adulthood there's a piece of technology that fixes nominally what it is that was problematic when you were first born. And these are, long-standing. I'm showing you Cheselden from 1728. Richard Gregory, one of my favorite scientists in the 1960s, studied a case like this. There's a movie from Oliver Sacks and a book and so forth about these kinds of things. We had the opportunity through Ione Fine's collaboration to study a person who's quite open about his case, Mike May. And Mike, at 3.5 years of age, lost one eye in a chemical explosion. He was blinded in the other eye. The cornea lens were destroyed. And he had unsuccessful corneal replacement in childhood. And then he lived for the next 43 years with light perception, but really no experience of contrast or form. By the way, Mike, for a period of years, had the world's record for speed downhill skiing. So he won the gold medal in the Paralympics, going at 60 miles an hour as a blind guy skiing. So he was a very courageous guy. And he decided at the age of 46 to try a corneal limbal stem cell transplant, which was done by Dr. Goldman at his center in San Francisco, and it took. And all of a sudden, contrast vision was returned to his retina. His electroretinogram was okay. He never really learned to see properly, and we were wondering, and we had the opportunity now, even though there are many cases like this, that was the expected outcome. We could make a measurement of what was going on in his brain that couldn't have been made before. And we could see that his maps were quite unusual in the sense that he didn't have the same foveal representation, and he had a retinotopic organization, but it really was not the same as others. And we went on to study Netta Levine in particular, went on to study other aspects of his white matter tracks that I'll tell you about later. So in summary then, for these first two parts of the talk, I wanted to say that plasticity is easily measurable and demonstrable in development. And you can pick it up, you can measure it with some precision, and it's a big effect, and they're quite remarkable. In the adult or early visual cortex, and I want to emphasize that, I'm not talking about the whole brain, I'm talking about the parts that I had mapped out earlier in early visual cortex, stability in the adult seemed to be the dominant theme. And that still seems to me true. There may be small amounts of plasticity that are below the resolution of what we could measure in these cases, because there's always something below the resolution you can measure. But the dominant theme was stability. So this raises the question, because we all learn to see new things, we learn to recognize new things, and one of the things that kids learn to see all the time are letters and words. And so the question is, well, where is that happening? How is the visual signal that comes in from the retina and primary visual cortex, how is that then learned by the reading centers of the brain, and where do those signals go? And I was, in any event, interested in understanding the connection between the visual signals that we encode and things that we think about and do and so forth. I am, after all, in a psychology department. So the history here is also really interesting and quite fantastic. I really trace the early parts of this to Carl Wernicke, who was the original connectionist. He was a person who thought that the white matter bundles that carry information from one part of cortex to another were the key to what made us human, and that it was not so much the cells or the nerves or the signaling processes which were preserved over all kinds of animal species, but it was rather the new connections that we formed and the way they formed and the way information was communicated that was really crucial. Jules Desjardins built on this with cases of reading and reading disorders that he and actually Wernicke conflicted about. Geschwin brought these back, and Elizabeth Warrington studied many cases like this. And so we walked into a field where people were thinking about connections. I have to say, when I first started looking at reading, the folks in the reading community largely said, well, what does vision have to do with reading? I mean, it's just not important there. So that was an interesting experience, but more about that later. So what is it that we knew about these reading centers? Well, starting in about 1990, we had pictures roughly like this from Samir Zeki, who's a great pioneer and wonderful anatomist and scientist understanding processing in the visual cortex. And looking in the ventral part, the bottom part of the human brain, we would have pictures that said, you know, color is kind of over here and words may be over there and so forth. And so we knew some stuff, and there had been some very nice papers by Kia Nobre and Greg McCarthy and Stan DeHaan and Laurent Cohn. And at this point in time, in parallel to what I've been telling you, many people have been working on the organization of the responses in ventral occipital cortex. And here's from a nice review paper from my colleague, Colleen Grospector, and Kevin Weiner, who was her student at the time, laying out a much more refined look of where different parts of the brain are responsive to faces and colors and so forth. And words are shown there, the little brown spot. So I look at these kinds of things to try to participate, and this is right adjacent to visual cortex. And we started to look at this and ask, can we trace how the visual signals are carried into the reading pathways, and perhaps as study children as they're learning to read, and see how these responses develop over time. And I'm only going to tell you a little bit about it, but the basic story is I believe that remains a good topic to study, even though I'm no longer focused on it myself. And it's something that you can see at individual subjects, in individual people, at spatial resolution, in experiments like this, where this is with Andreas Rauscheckner. And Andreas would have a person fixate here and present a word just to the right of fixation, and you can see the response in V1, V2, V3, the fourth visual field map, the ventral occipital maps, and all the way here into the visual word form area, in individual subjects, in experiments that last 15, 20 minutes, and so forth. And you can kind of look for these and study them. And we did have a look using the methods that I've been describing to you before, for example. I mentioned to you that we could measure these population receptive fields, and Karu Amano, Sergio Milan, would make measurements of these population receptive fields. Very easy to see these maps here in V1, but in the visual word form area that I was just showing you over here, these areas down here, they don't have maps. You look at them and you try to see the map, and we couldn't really find anything that looked like a map. But what we could do was say, if you collect up a number of different voxels from over here and see what it covers, well, yes, unsurprisingly, primary visual cortex covers a hemifield. Great. Well, you can do the same thing even when you don't have a map. You can take all the individual pixels and say, well, how much does it cover? And here is showing primary visual cortex, the ventral V2, ventral V3. When you look at the coverage of the visual field that's available in the visual word form area, it's actually quite small. So I hope you can see this is 5 degrees of eccentricity, 10 degrees of eccentricity. Eccentricity goes out to 80. A small part of the central region of the fovea is communicated into these regions. And so what's being trained is just that part. That's what gets there. And it's therefore a little perhaps it's not surprising that when you place a word in the far periphery, it doesn't get into the normal reading circuitry, so that the way it is you can read a word like that is to reason about it rather than to see it and read it naturally the way you do natural reading. In fact, we can make these measurements in individual subjects. There's 20 of them. And in fact, they have quite different, they're all small and they're all slightly lateralized, but they're different from one another. And perhaps these differences, you can't understand them on their own. This may lead to different eye movement patterns as kids are learning to read, or you might train different eye movement patterns and therefore get different types of coverage from these reading areas measured in this way with these tools. So as I said, as I've repeatedly said through this presentation, a lot of the things that happened from the 1880s to now have been based on the developments of new technologies. And the new technologies the new technologies in magnetic resonance are part of that story. And in fact, magnetic resonance is capable of making all kinds of other measurements in addition to the ones that I've been describing to you, such as the ones that Wernicke might have been interested in to understand the connections between different parts of the brain. And my lab did play a role in working out some of those techniques, and that was interesting, and that gave us the opportunity to try to understand which of the white matter pathways are carrying the signals between different parts. And I have to say, in thinking about what's next, if I was a young person in this audience and thinking about the signals in visual cortex and how they're communicated between different parts of the brain, I think I would be attracted to the idea of trying to build up system models of understanding how the signals in the retina and in V1 and in these different reading centers all interact with one another. They're not all feed forward, some are feed back. How could we model this? How could we understand this? How could we see this in the living human brain? And to that end, I'm a little old for this game, but I'm still in there punching. I'm looking at some other old people who are still in there punching also. And I have to say that, and it's a great privilege for me to be able to continue working with a younger person, to take the brainer on this, which is that we are doing our best to try to build such integrative models that start really all the way with the visual stimulus and three-dimensional renderings of spectral radiance fields, and understand how those are transformed by the cornea and the lens into photoreceptor excitations. And to build with Nicholas Guitar's models and collaborators from, such as Dave Williams' lab, models of how the retinal signals transform those so that we can get up to V1 entry, which parts of the responses can be explained by the peripheral, which by the central, and to keep going through the white matter pathways and in. And it's a wonderful challenge, and I hope we continue to be able to recruit fantastic students in these fields, and to keep our understanding developing, because there will be many applications of this as we broaden our scope. And with that, I want to just thank all of my students and postdocs and collaborators. This is actually a picture. I didn't know you were going to show the fat man and little boy thing, Dave, but that's a picture of Dave and me. Dave didn't tell you that during the movie Fat Man, when there's a critical moment for doing the countdown from 60 to 1 when they tested the atomic bomb, that was Dave's voice who did it, and he got it right on the first take. And I've been very lucky to work with these wonderful postdocs and students who are all running their own labs and having their own students at this point and doing all kinds of great things. And I particularly wanted to have a shout out to this rock group of clinicians who came to me from Japan, who really helped move a lot of this research in the direction of thinking back about clinical applications and so forth. So nice of you to be here and to work. And thank you all very much for this wonderful honor, for the ability to come and tell you this story. And thanks again.